So hello to everyone. I'm very happy to be here at this web conference organized by the EARN network. And today the topic will be about systemic sclerosis and the recent clinical trials. I'm Yannick ALLANORE, working in Paris at Cochin Hospital and the Université de Paris. Here my conflict of interest with regards to this presentation. And you know that on the cartoon here you can recognize the main pathways that contribute to the complex disease that is systemic sclerosis. So of course you can recognize the vascular component with the injury of the endothelial cells, the important immune and inflammatory component that is involving both innate immunity and adaptive immunity with the production of specific autoantibodies. And of course you recognize the very specific signal that occurs in systemic sclerosis that is promoting the fibrotic damages in various tissues that can be involved in systemic sclerosis. And I would like to show you the clinical trials and to position on this cartoon the drugs that have been investigated recently. And I would like to show you the main results about the immune component of the disease with the evaluation of rituximab, abatacept, tocilizumab, romilcimab. All of these are monoclonal antibodies. And I would like to also discuss with you two small molecules, the very recent data about Tofacitinib and the very important results reported with the use of nintedanib, and tyrosine kinase inhibitor in the context of systemic sclerosis. So let's start with rituximab. You know that rituximab is a monoclonal antibody targeting CD20, and FAI²R has reported an observational study that has evaluated the effect on skin and lung, and I would like to emphasize about the skin results, because using the registry we've been able to compare a large group of patients receiving rituximab, two patients having the same characteristics at baseline but not receiving rituximab. We've used a very careful design propensity score in order to select the best controls. And as you can see, if we compare the baseline visit to the last visit after about 2 years of follow-up, there is a larger decline in the MRSS from 22 to 14 in the patient receiving rituximab as compared to a reduction from 21 to 16 for the control patients not receiving rituximab. If we focus on the MCID, so the minimal clinically important difference, we can see that 23% are reaching this cutoff in the rituximab group as compared to 14 in the control patients. So it's a signal interesting regarding the skin changes. With regards to lung, I would like to show a study that compared the outcomes of patients randomized between cyclophosphamide versus rituximab. And all these patients had baseline scleroderma with interstitial lung disease, and you can see the variation in terms of FVC, GLCO, or the biomarker KL6. And for FVC, you can see that it's quite stable with regards to the treatment with cyclophosphamide, whereas you can see an improvement with a delta of about 20% regarding FVC for the patient receiving rituximab. And this was also true regarding DLCO, which is quite interesting, and this is also supported by the changes in KL6, which is a very interesting biomarker in the context of ILD. Therefore, we have encouraging data with regards to rituximab, and of course, you know, there is an ongoing randomized control trials in UK regarding lung, in CTD patients, and of course, we will look carefully at the results of this trial to try to see whether rituximab is really of importance for the treatment of the scleroderma patients. Then I would like to move to the tocilizumab trial. You know, the FOCUS trial, which is the phase 3 clinical trials, a large one, comparing 105 patients to 107 in the control arm, patients with early diffuse disease enriched for background inflammation regarding skin and also inflammatory biomarker. And you know the outcomes after about 1 year of treatment. You know that the patient in the control arm have a reduction of a little more than 4 units of MRSS as compared to a little more than 6 units in the patient receiving tocilizumab. And the difference is close to significance, but is not reaching the cutoff value. Therefore, the trial is considered as negative regarding the primary endpoint. You probably know that as a secondary outcome, lung was also measured in this patient. And it's very interesting to see that for the patient participating in this trial, the patient receiving tocilizumab have a very stable FVC during the 1 year of follow-up, whereas the patient in the placebo arm are losing FVC with a reduction of quite a large reduction, which is close to 5% of reduction of FVC. And this was true both for the patient having or not ILD at baseline. And I think the interpretation is that there is a preservation of lung function in the patient receiving tocilizumab. Of course, it was a secondary outcome. It's not a primary one, but it is raising the interest of blocking IL-6 in the context of very early inflammatory patients and with regards to the effects and the outcomes of lung. I would like to emphasize that the patient in the placebo arm are losing about 200 milliliters of FVC after 1 year of follow-up. And this is probably one way to identify the very enriched population that could be useful in the future for further clinical trials. So now I would like to move to the abatacept trial. So of course we know very well this drug, which is used in rheumatoid arthritis. And it was a phase 2 trial looking at 88 patients, early diffuse systemic sclerosis, baseline MRSS 22, mild lung involvement. It's really a skin-oriented clinical trials. And let's go directly to the outcomes and to the main results. It's quite similar to what has been observed with tocilizumab, with a loss of 4.5 units in the placebo arm as compared to 6.2 units for the patient receiving abatacept. And because of the sample size, it's not statistically significant. Also, although we can see that there is clearly a trend after month 6 during this clinical trial. So I don't know whether a final decision is taken today regarding the future of abatacept in systemic sclerosis, but you may see in the publication in Arthritis and Rheumatism that there is an attempt to identify and to select patients using the baseline transcriptomic data from the skin that could allow to identify patients who may be responder to abatacept. And I think it's a very elegant way, and maybe in the future it may be used for selecting patients who may be further evaluated for the effect of this drug. Now I would like to move to a very recent clinical trial. The data were presented at the last ACR meeting in Atlanta, and it is evaluated a very elegant component, which is a romilkimab, which is a bispecific monoclonal antibody blocking IL-4 and IL-13 at the same time. We know that in scleroderma there are many data to show the role of IL-4, and many data also showing that IL-13 can be a very pro-fibrotic cytokine. Therefore, there is really an interest of blocking these two pathways concomitantly in the patients. So it was a phase 2 trials, about 100 patients, quite early patient, baseline MRSS 21, lung involvement was modest during the trial, and one of the specificity of the trial is that it allowed the use of background therapy, which was mainly mycophenolate mofetil, used in about half of the patients. And you can see here the outcomes regarding skin. First of all, it's a 6-month trial, which is quite different from the previous trials I've shown you. And you can see that regarding the MRSS, the patient under the placebo are losing 2.4, whereas the patient receiving the drug under evaluation are losing 4.8 units of MRSS. And if we used the one-sided p-value measurement, which was pre-specified in the protocol, it's statistically significant, favoring the romilcimab and showing a significant effect. You can see here the two curves of the patient receiving the placebo, those receiving romilcimab, with a change after week 12, which is quite significant and quite large. I think of the most interest, you can see on this table the outcomes of patients receiving or not background therapy. You can see that in the patient receiving placebo, clearly there is a higher reduction when the patients are receiving background therapy. But of the most interest, if the patients are receiving romilcimab alone or in combination with MMF, you can see that there is a larger effect of romilcimab of this patient. The relative reduction is similar in the two groups, but clearly we get a larger effect when romilcimab is combined with background use MMF. And I think it's an interesting direction, and it's an important clinical trial regarding the compound and also the design that allowed the use of background therapy, which probably will be the template for the future. At the last Atlanta meeting, there has been a presentation, an oral presentation about Tofacitinib, a phase 2 safety study looking at these anti-JAK1, JAK3 inhibitors. Only 15 patients, most of them were receiving background therapy. It was a 6-month study, and the main result is that there was no unexpected side effect with the use of Tofacitinib. So there is no data regarding the safety for a so small sample size, but no warning signal with the use of JAK kinase Tofacitinib in the scleroderma patients. And I would like to conclude with the Sensis trial, which is of course the largest phase 3 trial ever done in the context of systemic sclerosis. So here we move from the skin field to the lung field with the treatment of patients with scleroderma and interstitial lung disease. You can see almost 300 patients included in the two arms and comparing nintedanib versus placebo. And in this trial, background therapy was also allowed, and I would like to show you the results. First of all, the main global results. You know these data that were published in the New England Journal of Medicine. The patients receiving placebo are losing about 100 milliliters of FVC after 1 year of follow-up, whereas the patients receiving nintedanib are losing 52 milliliters of FVC, which means a relative reduction of 44%, which is statistically significant. And you know that these data have been convincing enough for the FDA to take the decision to approve the drug for the treatment of scleroderma ILD. At the last ACR, several subgroup analyses have been shown. First of all, regarding the minimally clinically important difference, and these differences were calculated in the SLS1 and SLS2 studies, and you can see that with nintedanib, there is a higher proportion of patients having a significant improvement as compared to the patient treated by placebo, and there is a lower proportion of patients with deterioration as compared to the patient receiving the placebo. Of course, the most exciting data were probably those with the effect of mycophenolate in this trial. You can see that about half of the patients were receiving mycophenolate when they participated in this trial. Of course, this patient had some slightly different characteristics, more diffuse, higher MRSS, more altered FVC, but it was not a big difference. However, I would like to emphasize that these patients were treated by mycophenolate because the treating physician decided to keep on the drug for the patient during this trial. This means that we should consider this patient as at least being tolerant to the treatment or even being responders to the treatment with mycophenolate. So we should not overinterpret this finding. It was not a randomization of nintedanib with or without MMF. It's patient receiving background therapy, physician being happy with this treatment and adding or not in the randomized way nintedanib in this patient. But the results are the following. As you can see, they are quite interesting because they further support some effect of mycophenolate in this patient. As you can see, the patient not receiving mycophenolate are losing 120 milliliter of FVC as compared to 66 milliliter in the patient receiving placebo but having ongoing treatment with mycophenolate. And regarding the patient receiving nintedanib, again, the effect is higher in the patient receiving the combination. So the relative reduction is quite similar, but clearly the highest effect is obtained when nintedanib is combined with mycophenolate. The key point at this step is the safety, and there is no signal regarding the major or serious or severe adverse events. With regard to the more common adverse events, you can see here it's the comparison of the patient receiving nintedanib and mycophenolate. So 76% of at least one episode of diarrhea versus 75%, 31 versus 32% regarding nausea, and 23 versus 26% of patients having or reporting some vomiting during the trial. So no major effect of the combination as compared to the drug given alone. I would like to follow this with a few other data regarding some other subsets. The first one is the comparison of limited versus diffuse patients. So as expected, the patient having the diffuse cutaneous subtype are losing more FVC than the patient having the limited cutaneous diffuse. And you can see that the effect is probably slightly higher for nintedanib in the patient having the diffuse cutaneous subset. At the same time, you know that one of the secondary outcomes was skin, and it was interesting to measure skin changes in this trial, but you can see that overall the skin changes are really very, very mild, and clearly this trial was not designed to allow the measurement of the skin effect of nintedanib because the skin involvement was too mild in overall this patient to allow careful measurement and an accurate measurement of the effects of nintedanib on skin. So to conclude, I think we are now at a very exciting time because we have many clinical trials. We have many data. We get now the first positive clinical trials, and we are also improving the way we are designing the clinical trials. So we receive a lot of lessons from all these data. And my summary will be the following. I think we have encouraging data with the use of rituximab, and we hope in the next month or years to get even additional ones to support the use of this treatment. Regarding tocilizumab, the design of the trial has allowed the identification of a very specific subset of high risk of lung progression. Nevertheless, blocking IL-6 did not show efficacy regarding skin. So you probably know that Roche decided not to move forward for the registration of tocilizumab in that context, but it may be interesting for some other companies having other anti-IL-6 compound. Regarding abatacept, there was clearly some encouraging signal, but maybe in the future the patient selection may be improved, and we will see if molecular biology may be helpful in this context. I think that the phase 2 data about romilkimab are really very interesting, and the data deserve to move forward for a phase 3, which is considered by the company. JAK kinase is a very interesting and promising class of many drugs, and we have the very preliminary data in systemic sclerosis, but I guess that there will be further development with this compound or maybe even some other compound in the context of scleroderma. And finally, I think it's very positive for our patient that now drug is reaching the market, because nintedanib is going to be available in the near future for us to treat our patient, and we know that lung involvement is a serious concern for the scleroderma patient. Nevertheless, there are still some unanswered questions in front of us regarding the positioning of the treatment and the best selection of the patient who may receive this treatment, taking into account that overall nintedanib did show show that it could reduce the decline in FVC in patients with already established lung fibrosis. But it will be the matter of several developments in the future of very high interest for improving the care of our patients. So I thank you very much for your attention.